In the CUSP-ECR meeting on the 21st and 22nd of November 2023, I presented my work titled From in vitro to in vivo to humans, Microplastic Immunotoxicity. My name is Skrabanja and I'm a second-year PhD student in the UMC Utrecht. And my research project is about the effects of microplastics on the immune system. My focus of my research is mainly on innate immune cells, for example neutrophils, but also monocytes and macrophages. In my project, I have three main questions that I try to give an answer to. So my first question is, how do different microplastics affect neutrophil and monocyte survival over time? To answer this question, I developed an in vitro high-throughput assay where I can investigate neutrophil and monocytes, which I have isolated from blood from healthy volunteers, and assess their overall survival over time when I expose them to different microplastics. The microplastics I work with are derived from the both research consortia that I'm involved in, called Polyrisk, which is a European-funded consortium, and Momentum, which is a Dutch-funded consortium. From both these consortia, we received various forms of microplastics, different polymer types, and in my high-throughput assay, I can assess the overall toxicity. Based on the findings of my in vitro high-throughput assay, I will test the potentially most toxic microplastics also in an in vivo system. I injected the potentially most toxic microplastics intravenously in a mouse to study the overall microplastic dynamics over time, over a 30-day time period. And I will investigate organ accumulation, so where do those microplastics end up in an in vivo model, and I will assess if they are present or are they phagocytosed by immune cells, and are these immune cells activated? Do they have an activated phenotype? Lastly, I also intradermally injected microplastics to more specifically look on the long-term immune response when microplastics are present in a tissue. We're mainly interested in neutrophils because they are the first immune cell and the first responders in case a pathogen enters the body. Because a microplastic cannot be degraded like a bacterium or a fungal pathogen, we would expect that after phagocytosis, a neutrophil cannot phagocytose the plastic, or can phagocytose the plastic but cannot degrade it, which will probably cause a continuous immune cell response. Overall, I would really like to thank the both research consortia that I'm part of, but also the research group that I'm working on in the UMCU, which is the Kundeman Risiko Group. Thank you for listening.